So our planning really right now is not about if Zika it becomes locally transmitted through mosquito vectors in Texas, but rather when. We hope we can do everything that we can in cooperation and partnership with the national and local health departments and vector control agencies to absolutely postpone and maybe even completely prevent that from happening again, where we don't have local vector transmission in our state. However, we would be remiss if we didn't plan for the fact that that may happen. And that becomes, to me, the crux of the problem. It begins to be very challenging to understand what kind of response we will have in the event that we do see evidence that a case was acquired, most likely through local vector transmission via mosquito. The challenge then actually becomes that if you have, as it's, as the term of art is sort of used, if you have ongoing local transmission, vector transmission of Zika virus in a given area, then that really triggers your need to want to be able to test the pregnant women that may have come in contact with that particular area. So one of our main focuses has been to say, if we get local transmission, how can we define that in such a way that we're not just sort of opening up the floodgates on a demand for testing? And we had some reference to the testing earlier. And in a lot of ways, I think, to me, that is the biggest challenge. How would we meet the demand for testing of pregnant women if we begin to have local vector transmission within Texas? One of the things that there's special vulnerabilities, I think, in terms of Texas, and that's our proximity to the border with Mexico. I was down in the valley, as we call it, the lower Rio Grande Valley, not too long ago, and had a great visit. And one of the things to me that was really impressive is the fact that along that Rio Grande border, you're talking about a single set of communities, if you will, that happen to be divided by an international border. So it is a way of life there that we have hundreds of thousands of bidirectional crossings, legal crossings every day. People come into the US to work, to shop, to go to school and go back, and vice versa. So that's really something that I think is unique. And again, we know that there is, in fact, local vector transmitted Zika in Mexico, but we don't know everything that we'd like to know about exactly where that is and what kind of risk that presents. So we know that, again, that our climates here in Texas, if you look at a map of where the expected habitat is, breeding habitat is for the Aedes aegypti mosquito, it's pretty much the entire Gulf Coast, but then there's conveniently or inconveniently islands really around all of our major metropolitan areas in the state of Texas where we expect that mosquito to be. The mosquito, as you probably know, it feeds exclusively on human beings. It really loves to be around human beings, but it doesn't travel very far in its lifetime. And so that, to me, is one of the most other interesting facts about Zika. The virus is transmitted from person to person through an infected mosquito. From an actively viremic infected person gets bit by a mosquito, the mosquito takes that blood meal, the Zika virus then reproduces in their system, and probably over a period of about 9 or 10 days, when they're capable of retransmitting it to the next person. So that's how the infection occurs, but really the Zika is spread, if you will, geographically through the travel of human beings, through people migrating from one place to another, visiting one place to another. And obviously that's the case that we're seeing with regard to Brazil and other countries where we're worried about testing returning travelers. Some of the things that are challenges really specific to Texas: we don't have a statewide mosquito surveillance system. Texas is a home rule state, so that really means that the local entities, whether that be cities or counties, are really responsible for the first type of response. And so we've had a plan to communicate and reach out to our county judges, to our mayors and others to, before the season started, start talking to them about how to deny breeding habitat for the mosquito. Like I said, we'd love to know more about what exactly is happening in Mexico, and I hope that the CDC can devote some resources to help with that. And again, it gets kind of technical, I guess, but what really is the definition going to be for that sustained, or if you will, ongoing local transmission by a mosquito vector? We need to really have a good understanding of exactly what that means, because we're going to have to target our response to those specific cases and those specific areas. We'd love to know the risk in a statistical sense for pregnancy. We don't really have that basic information. And of course, that ends up being, in my opinion, what creates the real challenge for Zika, because this is, we know the devastating effects that this can have on the developing child in the womb. And naturally, every woman who's of childbearing age or who is pregnant is going to be concerned about that, and that concern will only intensify if we get local vector transmission of Zika. But right now, we don't have any even, if you will, statistical way of explaining what the risk might be. And so again, the risk, the danger to me in Zika is that women will want information in terms of their personal testing. They're doing serology-based testing is going to be the most likely means that we would follow pregnant women. And depending on how many cases we get, we don't want to be in a situation where the demand for that testing really completely outstrips our ability to provide it. We are cooperating with the surveillance system for microcephaly and for Zika cases, and we hope again that that is a great way that we will get that epidemiological information and will help explain the risk that a pregnant woman faces. So that's pretty much everything I know about Zika. It's been a real, I started this job in January, and this was, nobody said Zika till I walked in the door, so it's been taking off. I'm a little worried, and this part's a joke, so don't, I'm a little worried that I will lose family and friends to Zika because I bore people now with the subject. So thank you very much.
Thank you, Ken. And Brooke Courtney, welcome.
Great, thanks so much to NACCHO for organizing this session, and to everybody for coming out during dinner time. I really appreciate the opportunity to be here today to give just a quick update on FDA's key Zika activities. In public health preparedness, like I'm sure most of you here, I have worked on a number of unexpected issues, like getting a call one morning from my health commissioner when I worked at the local level that we needed to evacuate 50 horses and relocate them because they were being housed in a makeshift barn in an alley. And events like Zika really continued to surprise me. For example, I never thought that I'd be working on the regulation of mosquitoes that have been genetically modified, but here we are dealing with these new and interesting issues. Like others in the federal family, we at FDA are fully engaged with our partners across the US government, with the private sector, and also with the international community to help minimize the impact of the outbreak. And I just wanted to focus on 5 primary activities, primary areas of activity that FDA is working on with Zika. And I think they're supposed to be slides. If you can't pull them up, it's not a problem, but it might be helpful.
You're up.
Okay. So I'm just going to skip through these first 2. So currently, just to clarify, there are no FDA-approved, licensed, or cleared medical products available to prevent, treat, or diagnose Zika virus. Fortunately, we have a number of different authorities that we can use to help facilitate access to products that look promising based on appropriate scientific data, and we're prepared to use those authorities to the full extent possible, like we have with Ebola and with other emergencies. So the first primary area of activity is helping to protect the safety of the nation's blood supply and tissues for transplantation. So FDA is responsible for the regulatory oversight of the US blood supply. We regulate the collection of blood and blood components, and we work closely with other parts of the Public Health Service to identify and respond to potential threats to the blood supply or safety. And because the risk of transmission of Zika through blood and tissue products is such a serious concern, we're really actively engaged in this area of blood safety. So just wanted to give a couple of examples of what we've done. We have issued a couple different guidance documents. In February, we issued one focused on the deferral of blood donations from certain individuals, people who have been to areas with active Zika. And also that guidance significantly noted that blood should not be collected in areas with active Zika virus transmission unless it was tested with an investigational or approved screening test or had been treated with an investigational or approved pathogen reduction method. So this resulted in a very serious impact to Puerto Rico, which ultimately needed to import its entire blood supply for a short period of time until we were able to announce the availability of an investigational test to screen blood donations for Zika virus. So blood screening this test can now be done under investigational new drug application and IND in areas with active mosquito-borne transmission of Zika virus. And this is really significant for blood collection centers that have been, in particular in Puerto Rico, been very hard hit. So this has allowed Puerto Rico to begin to resume collection. We also issued a question and answer document related to that blood screening and deferral guidance. In addition, we're working on, with ASPR and BARDA and commercial developers on pathogen reduction technologies, particularly for whole blood and red blood cells, because there currently are no approved technologies for those. So those are also really important for the blood supply. And one thing, I guess two points about blood safety in particular is that we realize that very difficult decisions will need to be made if and when there is local transmission of Zika in the US mainland. And I know that we all share the same goal of protecting the public health, and that includes protecting the blood supply. So one issue is that there is a critical importance for states moving forward to have to establish an agreed upon definition of an area affected by vector-borne transmission of Zika for rapid reporting to CDC. And this information is really needed by blood collection centers throughout the US as the basis for activating the safeguards that we've laid out in our February guidance. And then the other is the value of blood donor screening test results as a really critical surveillance tool to complement public health reporting on the depth and the spread of the epidemic. We have, again, there are no commercially available diagnostic tests that FDA has cleared for detection of Zika. So we've been able to use our emergency use authorization or EUA authority to issue 2 EUAs for 2 different CDC-developed tests for Zika. And we're also working very closely with commercial product manufacturers to encourage them to develop tests. We've provided templates for them on how to request an EUA and what data is needed and so on. So we're working very interactively with them. And if it gets to the point where it's appropriate to issue an EUA, we stand ready to issue EUAs very, very quickly, assuming that the scientific data is there, and we've done that in the past. I know there's been a lot of concern about and questions about tests, laboratory-developed tests. And we have decided at FDA to require that clinical laboratories developing their own tests for Zika submit data to FDA for review for either approval or for an EUA, likely an EUA in this case. And that's really given potentially important decisions, life-altering decisions that families might need to make based on test results. It's imperative that the tests that we have out there and that are being used are accurate and are properly validated. And we've had some cases of sponsors not even using the most recent strains of Zika for analytic development and validation. So it's really important that that those tests come into FDA for review for an EUA. And thankfully, at this point, clinical labs have come to us and have been working with us closely on this. And when it comes to vaccines, again, there are no FDA-approved vaccines for Zika, but as we did with Ebola, we will do all we can to facilitate access to investigational products as they are developed, and we're actively working with NIH and ASPR and BARDA to advance the development of vaccines. So you get to vector control. Again, these novel and interesting issues always seem to emerge during emergencies. And as most of you know, insecticide resistance is increasing. So there is a lot of interest and emphasis on a range of different approaches for mosquito control. One of these approaches involves genetically modifying mosquitoes. So why is FDA involved in genetically modified insects? We don't have a statutory definition of the term animal, but an animal drug is intended to alter the structure or function of an animal, thus our current involvement in the genetically engineered mosquito from a company called Oxitec. And normally we can't talk about companies in this way, but Oxitec has allowed us to be able to talk about this. So you might have seen a draft environmental assessment and preliminary, what's called a FONSI, which is a preliminary finding of no significant impact related to this mosquito, but it's part of our regulatory process to get to the point where Oxitec wants to conduct a field trial in Key Haven, Florida. For them to be able to do that, we have to issue these documents and open them up for public comment. So we are in the process now of accepting those public comments, and based in direct response to public requests, we just extended that period of comment, public comment, to May 13th. So if you're interested in submitting comments, looking at the draft environmental assessment and the FONSI, we encourage you, reach out to me or look on the FDA website, and it's pretty easy to find. The way this mosquito works is that it's grown in labs, and genetically engineered males will then be released every few days to mate with wild females, and then their eggs will hatch, but no adults will result from the mosquitoes. And the intent is to suppress the mosquito population. It's not to prevent disease transmission. And again, the field trial can only occur if FDA issues a final environmental assessment and a final FONSI. So just to reiterate, at this time, FDA is not approving the Oxitec application. At this time, we're still going through the process. I've heard a couple of people in recent meetings talk about how FDA approved the mosquito for use, and we haven't yet. We're still going through the review process. But Oxitec could use those results from the field trial in an application toward approval. And there's also been some talk that this mosquito is approved in Brazil, but it also has not yet been approved in Brazil, although has been widely used there. And then in my remaining 19 or 15 seconds, I guess I'm over now, technically, we are working on, it's like the numbers are going up, not down now. We are involved in the monitoring of fraudulent projects. So unfortunately, during outbreak situations, a number of fraudulent products come to bear that claim to prevent, treat, or cure a disease. And we dealt with a lot of this during the Ebola response. So we're monitoring for fraudulent products for Zika, and we encourage any of you, if you hear of any such products, let us know, and we will handle that with the developer in the appropriate way. So thank you so much for your time, and turn it over.
Thank you so much. Next, we'll call Scott Becker to the... Thank you, Scott.
Thank you very much. So first of all, I am not a laboratorian, so this will not be very technical. So for the laboratorians in the audience, sorry. What I'd like to do is to talk a little bit about the lab response. You've heard from some of our other colleagues about some of the challenges with testing and some of the challenges as it relates to Zika response, and testing is certainly one of those. There's scientific challenges, there's any number of challenges, but it's not only about challenges, we're also going to talk a little bit about opportunities. So I'll talk a little bit about the testing capabilities in the country, what our organization has done in collaboration with many of the partners up here, and then some of the ongoing needs and challenges. And here's a hint, one of the ongoing needs, money. So it was referred to, and I think the folks in the administration have done a great job in trying to make the case to Congress, but it's going to be up to us, all of us, to actually raise our voices and let Congress know we really need this money. That the response for Zika does not equal the response for Ebola, does not equal the response for flooding in Southeast Texas, that does not equal response to Elizabeth Kingia or whatever else is going to hit us, because it will. I have no doubt that the new normal is that there will be an EOC operational most times of the year. So let's just get ready for that. So the current status was shared earlier. The point here is that a lot of it is the transmission, locally acquired cases are happening in Puerto Rico. I was recently in the Marshall Islands. Upon landing, I was given a Zika prevention kit. I thought that was terrific. In the US, it will, and again, it's not if, it is when it becomes locally acquired, things will spike. We're going to go through the same experience we did when Ebola was in West Africa, and then it was here. Why is it always Texas? I'm not sure. But, so let's go over a little bit of the tests that are available. So the FDA did approve an emergency use authorization for the real-time PCR and the Trioplex. Okay, the Trioplex came about a month after the original EUA for the real-time PCR, and the Trioplex covers Zika, dengue, and chikungunya because they're so closely related. There is also a lab-developed test, a real-time PCR, specifically for Zika. That's the molecular, and I'll talk to my colleague later from FDA about what that means. Now, I believe the real-time PCR is simply because of the extraction equipment that's used. I'm not quite sure. But the key for PCR is that the molecular tests, or any of these tests, these are all both PCR, is useful within 4 to 7 days of the onset of illness. So if you imagine a point in the future, if there is locally acquired transmission, you are going to want to get those pregnant women into a facility that can send a test or can test themselves. We're not at the point where anyone in the US can test except public health laboratories or those laboratories affiliated with CDC's LRN. The second test available is serology, and there's also an EUA. It's a MAC ELISA. This has cross-reactivity to other flaviviruses. So it's a good test. It's not a great test. And then the last test that's available is called PRNT, plaque reduction
Neutralization.
Okay, PRNT. I can't hear them. So PRNT is the confirmatory test. And that test is only available in 4 labs in our country because we gave that test up a long time ago. We gave that test up like we've given up a lot of other tests that were classic microbiology procedures. So that's a rate limiting factor for us. So here's where we were in March, about 5 weeks ago. The red indicates the laboratories that were not anywhere on the scene for Zika testing, for validation. Okay? The teal color are the labs that had already validated, and the others were in process. So here's where we are now. There's only one. We are following up with South Dakota to understand. We believe that they have an agreement with a neighboring state. And they're also so far north it may not be so much of an issue other than travel associated. But we are going to follow up with them. But you can see many, many more are in teal at this point. And this says as of 4/19, but I can tell you that Virginia, we have Virginians here, your lab was validated, they passed the validation and they are able to test for Zika using the CDC assays. So here's what APHL did. Interestingly, internally we were saying, hey, Zika's coming, we need to do something about this. So we got our team together, our preparedness and our infectious disease team, and we started to plan in advance, much like we did with chikungunya. And then on the 18th of January, I got a call, and it was from the Canadians, and said, can you join us? That was before our own country got started. Now, there may very well have been discussions internally within CDC and other agencies, but we were asked first by Canada. So we did share the testing guidance, and we jumped, along with CDC, jumped ahead right into the fray and got some lab alert calls going. We got 50 state calls. We brought people together with the lab team that's been assembled at CDC. We stood up a Zika lab testing working group, which is a technical working group of our organization to advise, guide, and work with CDC. We did some early surveying, and you can see the rest. But the key is that we've been educating our members for every change that's come along, including any of the new guidances. And most recently, we have contributed along with CSTE, ASTO, and NACCHO to a follow-up survey for states looking at what the implication is going to be of the PhEP redirection, because we know that there's going to be a big implication. This is just a simple schematic of the incident management structure. I like to call it ICS light, since we don't really organize the way CDC does. But it's based off the same ICS system, and it's actually worked very, very well for us, where we have a public information officer, policy officer, science officer, along with our incident commander who's here, Chris Mangle, who heads up our preparedness and response program. So incident management is one of the things that we've continued to bring. Even though we're a national association, we also feel like we are a response agency. We help to coordinate and collaborate with the laboratories across the country, with partner organizations, with the federal government. Provided technical assistance in many forms, provided assessments, and then collaborated in partnership. So this is an example, but this is just one of many examples. Our public policy work, I think you all often don't hear enough about, and I don't mean just APHL. I think I am also referring to ASTO and NACCHO and CSTE. The folks that are in DC who work on public policy for you all are doing a phenomenal job. They're the ones who are not hamstrung by their jobs to go and say to either the administration, to the White House, to OMB, to Congress, here's what we need. Here's what's going on. This is the impact of you not acting, or this is the impact of you acting in a certain way. And then, of course, media, and we've all done a lot of media throughout this. So let me spend just a second on the public health laboratory needs. Well, the first need that we discovered is you can't develop the test unless you have convalescent sera. You need materials to develop the test. And we didn't have patients here. And it wasn't really feasible to go to Brazil and try and get their convalescent sera because they were using it. We had to figure out ways, and that wasn't in any means APHL, that was really CDC. Limited availability of sera, that is an ongoing thing. I mean, folks, at the beginning, we needed buckets of sera to make the number of tests that are needed for our country. The guidance for lab-developed tests, most of the test kit is actually a kit, but it also could be construed as a lab-developed test, meaning the labs get all the materials they need, put it together. It's a special recipe, if you will,